Hi everyone, thanks for listening. Today we're looking at one of the biggest challenges in central nervous system drug development, crossing the blood-brain barrier. So over the next few minutes, we'll briefly cover why the blood-brain barrier is such a challenge and how VHHs could support future central nervous system therapeutics and imaging approaches. So the blood-brain barrier is a highly selective interface that protects the brain by tightly controlling what can pass from the bloodstream into the central nervous system. Is formed by specialized endothelial cells connected by tight junctions which create an extremely restrictive barrier. Its primary role is to protect the brain from toxins and pathogens circulating in the blood, whilst at the same time it is not completely closed. The blood-brain barrier carefully regulates the passage of essential molecules like glucose and oxygen, as well as key nutrients such as amino acids. It also plays an active role in maintaining brain health by facilitating the removal of waste products back into the bloodstream. So overall, the blood-brain barrier is critical for maintaining a stable environment, or homeostasis, which is essential for proper brain function. But from a therapeutic perspective, the restrictiveness of the blood-brain barrier is a major challenge in treating central nervous system disorders. So, the delivery challenge. Nearly all large biologics, such as antibiotics, peptides, and viral vectors, and even most small molecules, can't cross the blood-brain barrier. In practice, this means that even when we administer therapeutics systematically, very little can actually reach the brain. So, for example, monoclonal antibodies often achieve less than 0.1% of the injected dose in the brain tissue. So, to compensate, we rely on high-dose regimes, but that can introduce cost, safety risks, and side effects. There are also more invasive approaches, like focused ultrasound, to temporarily disrupt the barrier, but these come with complexity and scalability challenges. So overall, the blood-brain barrier remains a fundamental bottleneck, and we need to find smarter ways to get drugs across. So to begin to overcome this delivery challenge, researchers have looked into exploiting natural transport mechanisms across the blood-brain barrier. So small molecules can sometimes pass via passive diffusion, but this doesn't work for large biologics. Nutrients like glucose use carrier-mediated transport systems, but these are highly selective and not easily repurposed. The most promising route is receptor-mediated transcytosis, or RMT. This is how large molecules like transferrin or insulin naturally cross the blood-brain barrier, and it's the pathway most commonly used to shuttle therapeutics into the brain. The different transport systems can be seen in the figure on the left of this slide. So even this approach does have limitations, such as receptor saturation and competition with natural ligands. So this is where VHH antibodies come in. So VHHs are single-domain antibodies derived from camelids, and they're about 1/10 the size of a conventional antibody. The small size gives them several advantages, including better tissue penetration, more efficient transport, and greater flexibility in engineering. We're already seeing 3 key application areas emerge. So the first is therapeutics, where VHHs can directly target disease-relevant proteins in the brain. Second, imaging. So radio-labeled VHHs can rapidly penetrate tissue and generate high-contrast images. And third, gene therapy, where VHHs can be incorporated into delivery systems like viral vectors to improve targeting to the central nervous system. But what really sets VHH apart is their modular design, which is opening the possibility to a range of strategies for crossing the blood-brain barrier. So how can VHHs help with central nervous system delivery in terms of their modularity? So on the left here is a simplified example of a modular VHH construct. So one domain here acts as a blood-brain barrier shuttle, which can be linked to a second functional module or payload. So importantly, that payload can be highly flexible. It could be another therapeutic VHH, a protein, a nanoparticle, or even an oligonucleotide-based therapeutic, such as single-stranded RNA or antisense oligonucleotides. So on the right is a conceptual example of how these constructs may function at the blood-brain barrier. So first, the shuttle VHH can engage a blood-brain barrier receptor, such as the transferrin receptor, triggering internalization and transcytosis across the endothelial cell. So once released, the payload can then engage its intended central nervous system target, in this example, an amyloid beta plaque. So this type of modular architecture is attracting significant interest because it offers a potentially tunable platform for combining blood-brain barrier transport with therapeutic or diagnostic delivery in a single construct. So while many of these approaches are still at the preclinical stage and still face a number of challenges surrounding transport efficiency, intracellular trafficking, and systematic pharmacology, they highlight the engineering flexibility of VHH. So to summarize, the blood-brain barrier remains one of the biggest challenges in central nervous system drug development. While receptor-mediated transport offers a viable route across it, success depends on the right molecular format. VHH antibodies combine small size, stability, and engineering flexibility, making them uniquely well-suited as blood-brain barrier shuttles. And importantly, they unlock multiple applications from therapeutics to imaging to gene delivery. So overall, VHHs represent a powerful platform for finally accessing targets on the other side of the blood-brain barrier. Okay, that's everything. Thanks for listening.